Thank you all for coming to the late pandemic reboot of the Anthropology Colloquium series. My name is Paul Bravo, and I'm a professor in the Anthropology Department and chair of the Colloquium series. Our speaker today is Melanie Beasley. Melanie Beasley is an assistant professor in the Department of Anthropology at Purdue University, where she's also a faculty fellow in the Cornerstone Integrated Liberal Arts Program. Professor Beasley received her PhD from UC San Diego in 2016, and then took a postdoctoral fellowship at the University of Tennessee, Knoxville, where she did research at the Forensic Anthropology Center, also known as the Body Farm. As a biological anthropologist, Professor Beasley studies human-environment interactions of the past and present, where the word past means the last 4 million years. She's carried out research in East Africa, California, Jordan, and Italy. In addition to her work in biological anthropology and bioarchaeology, she uses stable isotope analysis for forensic applications to aid in the identification of human remains and determine the time since death. I'm assuming these are parts of the talk today. So let me just quickly thank Dr. Beasley for driving up today from Indiana in pretty horrible weather in order to share her work with us. So please join me in welcoming Dr. Beasley to our campus this afternoon, where our talk is entitled "The Tale of Two Stable Isotope Projects: How Development of a New Forensic Anthropology Method Tests a Paleoanthropology Hypothesis."
Thank you so much for that very kind introduction, and thank you for inviting me and that I get to be your first in-person speaker right here since 2019. So this is what an honor. Thank you so much. And I'm so excited because I actually started at Purdue in 2019. So I feel I haven't gotten to know all of my local new Midwest colleagues here in the area because as Paul mentioned, I'm from California. I did actually all of my education and training in California. And then I moved to Tennessee to go work at the Body Farm for my postdoc. And so this is my first time in the Midwest. So this is wonderful to start to get to know all of the other local anthropology departments. My talk today, I did want to say there will be a couple of pictures of human remains. These are all donated individuals at University of Tennessee. Of course, all of that work cannot be done without the generous support of donors, not in the financial sense, but in the actual, greatest gift that they can give, which is of themselves for this type of research. So I do want to always start and acknowledge with that. Thinking through in this talk, I have given this in various iterations, but the pitch that I wanted to kind of frame it in today is really for students and thinking through, this came out of me just finishing my dissertation, hearing a talk at the end of my dissertation time and thinking through a new postdoc project in a way that I could do kind of two very different projects, but how the same data and the same kind of research design could actually inform these two very vastly different areas of forensic anthropology and paleoanthropology. So the research that, things that I'll be talking about, I always want to kind of highlight the new lab that I have at Purdue, which is the Isotope Ecology Research Lab, known as the Beer Lab. Yes, that was done intentionally. And so a lot of the work, those samples were collected in Tennessee. I had a lot of great undergrads who've now helped me over the last few years in processing these samples. The work that I come at, and Paul mentioned this in his very nice introduction, is as a biological anthropologist, I'm interested in these broad questions about human-environment interaction. And I have worked in time periods when the environment is acting as the greater shaper to past human populations in East Africa. I've worked in bioarchaeology, specifically a lot in California, in looking at zooarchaeological questions of when humans were changing their diet breadth and therefore acting as greater shapers to their environment. See, I say zooarch, and the zooarchaeologist is like, yeah. And then also in this time period of forensic anthropology, when in our modern world, the modern environment someone is living in is imprinting in their biological tissues. And so I look at all of these questions very specifically through this one method of stable isotopes. And I will give go too heavily into the details of isotopes at this moment, except to say in general, so that we're all on the same page, stable isotopes is a way that you're looking at these elements that have same number of protons, different number of neutrons, and you're comparing a heavy isotope to a light isotope of whatever element of interest you're in. And so you get these delta values where you get this ratio relative to a standard, and anthropologists have used different isotopes, strontium and lead, oxygen and hydrogen, carbon, nitrogen, and sulfur. Regularly, these isotopes are turned to by anthropologists to look at questions about geography, migration, paleoenvironment, past behavior, dietary behavior specifically. And so this is kind of in this toolkit that anthropologists have. We can do this very well deep into the past because these isotopes are recording in different biological tissues. And so biological tissues are going to record essentially a snapshot of information about whatever isotope you're interested in. So if you're interested in, say, where someone is from, that tooth enamel, which doesn't remodel, is going to record those values when that tooth developed. The bone is going to remodel throughout your entire life. So that bone is going to be an average over the last decade of life of where you've been and what you've eaten and what you've consumed. Whereas something like hair and fingernails, which are not just for modern context, but a lot of mummy work is also done in archaeology, where you have very good site preservation. You can do work on hair or fingernails where this is going to represent snapshots of the last week's or months worth of time. So anthropologists, in thinking about these different types of research questions, we have a suite of different biological tissues with which we can turn to use. And I like giving this kind of retrospective over the last 20 years of presentations at the AABA, SAA, and the American Academy of Forensic Science meetings. This is specifically data from the anthropology section, that though forensics has lagged behind in the uptake of forensics as or isotopes as a method in forensic anthropology, you can see definitely that there are huge upticks over even just the last 20 years and how regularly stable isotopes are being used by anthropologists to ask different types of questions. And so where I wanted to start today's kind of story is thinking about my history in stable isotopes and the fact that I came to this method with really an interest originally in paleo diet reconstructions. This was the first time that I learned isotopes, doing paleo diet reconstruction with California individuals during my work at Chico, and thinking through how people regularly do these paleo diet reconstructions with material material culture by looking at faunal material or floral material, artifact assemblages, reconstructing the paleo environment in some way that, potentially what people are using stable isotopes as a supplement to these other methods. But of course, one question in these, how do we reconstruct paleo diet, is, are we actually reconstructing what is being consumed? We have limitations based a lot on what is preserved and what is recoverable in all of these different types of methods. And within stable isotopes, you can get a value, but that single value for an individual is really an average over, say, a decade of their life if you're looking at bone collagen. And so all here, are we actually looking at what individuals are consuming? And so when you think not just as paleo diet, but what is on the paleo menu, it becomes a lot more complex for humans rather than when you're looking at these other, let's say, kind of simple mammalian species where maybe you only have one or two dietary inputs. And so you can reconstruct with isotopes those contributions, say, to the protein sources in their diet, which is regularly done with collagen isotopes. So I would argue, and others have argued as well, that we need to expand our thinking about beyond just this, how are we looking at a paleo diet reconstruction, to really thinking what's on that potential paleo menu, and of that paleo menu, what is potentially being missed by the ways in which we're doing diet reconstruction now. There's a great paper that came out just in the last couple years looking at kind of some examples of food processing methods in terms of looking at meat drying in various different cultures. And you can even see these lovely maggot larvae that is on this meat. This will play into the larger story when we're ongoing, as well as food preparation practices. And so these are things that are not often considered as part of that paleo menu and how they might impact isotope values. And so today, and thinking through some of this in terms of what should we be considering for the Neanderthal paleo menu, this truly becomes a complicated isotope story, because when individuals were first looking at this paleo diet reconstruction of Neanderthal sites and looking at whole entire food webs, so looking at the herbivores and the carnivores and then where the Neanderthals fell, back to the 90s and the early 2000s, lots of different sites, oops, I'm gonna kind of run through a couple of these. I'm gonna show the Neanderthals are all in the red circle around these different sites, where continually you have this perspective that they are the high trophic level species within that food web. They have those highest nitrogen values. And so the interpretation off of this was essentially that Neanderthals, through these nitrogen values, were these top predator carnivores, and that they were eating at this very high trophic level carnivory within this paleo habitat that they were living in. But of course, though that story emerged in the 90s and early 2000s, a lot based on this nitrogen data, you then had additional papers that started to come out to kind of expand that complexity of it not just being all high meat hunting carnivory that they're consuming, but looking at things like evidence of marine mammal exploitation. Amanda Henry's work, looking at a lot of, and her group that works on a lot of these plants and cooked foods and contributions to Neanderthal diet. The star of today's talk will be this piece by John Speth that came out in 2017 about the consumption of putrid meat and fish in terms of thinking through, is this a key missing part of this Neanderthal dietary tale and these high nitrogen values? As well as that same spring, Royer et al. Came out with looking at how isotope values are impacted and altered based on cooking. And so if we take just first this Royer article, very simply, this is just looking at lots of different meats that then they cooked in 3 different preparations, and they looked to see if there were any changes in the isotope values in those cooked preparations. So 0 means no change, and so you don't have a lot of significant change going on with cooked meat. And I can tell you interpretively, isotopically, these values, a change of a couple of tenths of per mil is not interpretively going to be a big difference. But in some cases, the fish, that is a different story. Now, this story is not a story about fish today, but I'm saying, I'm making the argument to the end that we have to continue to be very wary of thinking about simplistic reconstructions of paleo diet, because these cultural overlays of cooking, of food processing, is potentially changing isotopic values that we're not considering when we're looking at baseline animal values to reconstruct a paleo diet in prehistory. Now, what Speth did, to me, in my mind, is one of possibly the most beautiful pieces ever written that focuses on putrid meat and consumption of maggot larvae, because what he's done in this 2017 paper is made an argument that we are missing some of these dietary inputs. And he went to the ethnographic literature of a lot of the Arctic populations and a lot of those early contact ethnographies to find quotes and evidence of the consumption of what we in a Western thought would think of as putrid or disgusting, or we're not going to eat that in some way. And yet this was not that local perspective. Anyone like stinky cheese? Some people are like, anyone's like, oh no, that smells like mold. Nope, I'm not gonna eat that. You know, what are you culturally used to? What Speth found is that in his research of not only the food science literature, but also delving into the literature of forensics and looking at the ethnography, is that there's a lot of benefits from eating partially, essentially digested food, and that digestion is not through consumption inside the body, but rather that putrefaction process that's going on outside of the body. Because if it's decomposing and breaking down in beneficial ways, there's no need for fire, no need for fuel in these Arctic populations. And there are benefits such as lactic acid bacteria, which is preventing certain pathogens, producing certain vitamins that's going to preserve vitamin C. There are these benefits from this method. And in fact, he found this example that botulism, which was a great concern of Westerners, Native groups didn't experience that until the Westerners came in and said, hey guys, you're doing this wrong. This is unhealthy. Here is some plastic containers. And only then, by not doing it in their traditional cultural practices, were there these pathogenic issues. So what Speth found, and I only want to kind of highlight some of these wonderful quotes. It's the most beautiful article. He has like 5 pages of these ethnographic poll quotes that he's compiled. And these are just some examples of the whole is then, so this is referring to the carcass, the whole carcass is then deposited under the nearest cleft of rock and stones and are built round. And they seldom use meat till it is rotten. They keep it in their huts, unexposed to air. And so there's this idea that putrid and pathogenic are not synonymous. And so whether you call it fermentation or spoiled or putrid or rotten, that's actually coming from our kind of Western bias of some of these foods, and it's not yet rancid. So these are culturally specific delicacies, and so if you're growing up in that culture, you're like, oh, mom and dad are eating that. Oh, I'm going to eat that too. That's lovely and delicious, and that's yummy. And so you're not going to have that kind of consumption of putrid meat. Like you don't have that gut reaction of disgust because that doesn't develop until about the age of 5 to 7. And so if you're growing up and observing that, that's not what is being seen. See, the idea set forth is that how do we test this consumption of putrid meat? Well, John happened to come and give a talk, just like I'm coming and giving you guys a talk, in 2017, and my postdoc application is in at the University of Tennessee, and he gives this talk, and he talks all about how that the nitrogen is one way in which this can be looked at, is that these high nitrogen values in Neanderthals are actually coming from this putrefaction process because these volatile organic compounds are being produced during putrefaction. And so the process of this is that, say you have your dead carcass, you have some distribution of these heavy and these light nitrogen isotopes, and over time, that light isotope of N14 is going to preferentially be making these volatile compounds. Those volatile compounds are being released, and so over time, you have more of the heavy N15. And this relates to this ratio that you're looking at of N15 to N14. So through time, those nitrogen values should increase. This means that maybe these high nitrogen values in Neanderthal sites that we've been seeing are not just an indication of being a top carnivore, as we've seen from other evidence, they're eating other things, plant foods are contributing, but maybe we can actually get at this food processing technique that they're using, because these values continue to go up. So why Tennessee? Well, so I'm sitting there in the audience. This is February of 2017. John's paper hasn't even come out yet, so I haven't read it. It's just, he's talking about it in a talk. And afterwards I look at him and I go, John, I could do this. Like, I could not do this with animals, but I could do this with people. And then we could also look at these values as a postmortem interval estimator. And nobody's done that. And so this is why Tennessee was perfect. And then I was, crossing fingers in February that I was going to get this postdoc. And luckily, later that spring, I did get the postdoc and immediately was an email to John. Oh my gosh, I'm going to change my postdoc project, and this is going to be one of the main projects that I'm now going to test while I'm there. Because the great and beautiful thing about, lost my ability to click. Watch it like, it is thinking, it is, whoa. Nope, you go away. All right, where'd you go? Okay, well, we might have to use that clicker now. The great thing about Tennessee is they have the Forensic Anthropology Center. So this was the first, as Paul mentioned, affectionately known as the first body farm that was established in the world. So in 1981, Bill Bass established this donor program where human decomposition could be studied, as well as tissues then were collected. Hair, fingernails, bones, teeth. The skeletons go into, after being at the facility, they then go into a curatorial skeletal collection for further study. And to date, since 1981, there are about 2,000 donors that have come through the doors of the Forensic Anthropology Center. And annually, there are about 200 donors or so at this point every year. This has exponentially, obviously, grown since the 80s. But on average, there's somewhere in the neighborhood of about 200 donors that are at ARF at any one time. So I took what is known as the Haslam Postdoctoral Fellowship. And the wonderful thing about this is that this is a private fellowship foundation from the Haslam Foundation. They fund this research, and so this is not a postdoc where you are like getting onto somebody else's project, but you basically propose what you want to do. And so they've had a geneticist. Mike does everything. I just kind of describe him as a statistician, but Mike does a lot of different things. They had me as an isotope person, and currently they have an entomologist who is in this postdoctoral position. And so the wonderful thing about this is I got a chunk of money just to fund and do research. So I was able to design my own projects with isotopes that were going to also help forensic anthropology. But in the back of my mind, the whole time I knew I could test this high nitrogen tale, essentially, about the consumption of putrid meat. So let me pause on Neanderthals and switch to, okay, so I get to Tennessee, I have this postdoctoral fellowship, and I'm thinking about all the things that I can do with isotopes because strontium, lead, oxygen, hydrogen, and carbon have all been shown to contribute to various information about the biological profile of someone. But the big question, well, trauma, you know, if we think about these as different areas in which you can do forensic anthropology research broadly, trauma is probably not going to be an area that isotopes can contribute to, or at least I can't think of anything of it yet, though it would be exciting. But postmortem interval, nobody's been looking at this with isotopes. So I arrived in 2017 saying, okay, well, we can look and see what nitrogen specifically can contribute to this story. And so I designed this study where I placed initially as a pilot study 10 donors in the winter in January, 10 donors in the summer in July, to then follow their decomposition and sample their muscle tissue and then sample the associated maggots that arrived on the bodies after that. And this was all to look at just using solely nitrogen, can we estimate post-mortem interval, how long someone has been dead?
Oh, can I ask you a question?
Absolutely.
You said that fingernails, et cetera, were curated?
Yeah.
That's before they're put in the ground?
Well, the individuals at the facility are not all put in the ground. Most donors are initially put on the surface. There are donors that do end up getting buried for a lot of the short courses that they do. Yes, at intake, when individuals come, there are samples of hair samples and fingernail samples that are collected upon arrival, with the exception if they don't have hair, or sometimes fingernails are not available because we do not remove.
But that's supposed to give you a baseline.
Yes. And so, and that is part of this study. Like the baseline is based off of that. In this study, I'm placing these donors and the initial baseline is muscle tissue. And so I'm relating it all to that first muscle tissue sample. I also did the isotopes of the hair and the fingernails because in a forensic case, you wouldn't have that initial muscle tissue sample So you have to relate it back to some other biological tissue that could be used as that baseline. The other thing about donors, as you know, I just mentioned, they are placed on the surface initially. They're placed under black plastic, usually for just kind of, unless the study requires that the black plastic isn't covering them, this is out of respect for the donors. And so therefore, one study I initially did in these first 10 winter donors was left part of the body covered, basically one leg covered with plastic, one leg not covered with plastic, to see whether or not the plastic was impacting that nitrogen change that I was looking for in the muscle tissue. And just doing a simple t-test, no, it did not change at all. So there was not a problem with me collecting muscle tissue from donors that are covered with plastic. So a couple of the graphs that I'll be showing just to kind of look at how this runs. Some indicator of time. This is number of days at ARF. There's also going to be some charts that have ADD, so accumulated degree days. But some value of time is going to be on the x-axis. And on the y-axis is this big delta in 15. This big delta is a difference between two small deltas values. So essentially, I have the difference of whatever that day that I collected the muscle tissue value on, and that's starting out that first day that the sample was collected when they were placed at ARF. This is just an example of kind of what my traditional winter donor patterns look like, that there's some period of lag where there's not much change, and then through time you get, somewhere in the neighborhood of about 2 to 3 per mil increase in those muscle tissue values. This is a pilot study, right? So this is just essentially a proof of concept with 10 individuals that yes, indeed, we do see this as predicted, as hypothesized, increase in the nitrogen values of muscle tissue over time. This just shows you this now. All of the black dots are the averages of the winter placement values that are binned into these 10-day groupings, essentially. And then the summer values, because summer decomp in Tennessee goes much faster. That, yes, there's a little bit of an uptick here, but not very much, and the muscle tissue in the summer is a lot more difficult than the winter values, which increase significantly above, usually about 2 per mil, as I said, but there is a wide range of variation. Statistically, this is where you start to see this change essentially after a month, after about 30 days of being placed in temperatures of around 43, 44 degrees Fahrenheit. And there is this change in this average daily temperature that basically as it gets warmer in the winter, that yes, you do start to see this change in this period. It'd be great. Obviously, this is one study, one location. The idea would be to take this to other human decomp facilities and try to test this in different types of environments to see how does, like, just 3 hours north of you where frost is, how much stability in that nitrogen happens when you have a lot of snow. There's not a lot of snow in Tennessee. This is just to kind of show you here with the muscle value spread. So this is all of them plotted next to accumulated degree days. The reason this is important, not all donors come to the facility, like say immediately. Sometimes for various reasons, they're at funeral homes for a while. So this is to try to adjust for this period where donors might have been in refrigerators at funeral homes for a longer period of time before coming to Tennessee, before being in a freezer, because I held until we had 10 bodies to all put out at the same day so that we could control for environment as best as I could. But you can see, yes, you have this uptick. Yes, you have some outlier values. Ran those multiple times. Those are real values. So you do get this spread and this difference in these values. One of the things to think about in terms of this larger kind of Neanderthal question is, well, what about body size? Obviously, humans are not going to approximate the size of terrestrial herbivores that I'd be applying this to in a Neanderthal model. Well, at least, in terms of having a spread of muscle tissue, there is some evidence that you're still seeing this general pattern of uptick in nitrogen values. Would it be ideal to be able to do this with larger bodied mammals, you'd be surprised that it's very hard to convince people to let you put out a bunch of cows on a field and decompose them. There has been one study with beavers, and this same, actually by someone in forestry or agriculture at Tennessee, doing a different project, and she looked at beavers. This same nitrogen pattern kind of follows with the beavers. So now what about the maggots? So we talked about the muscle tissue. There's some, it's looking good from the pilot data that yes, these nitrogen values have something to do in terms of this postmortem interval change in these nitrogen values. But I'll tell you, star of the show, never thought I'd say it, are the maggots. When we turn back to the ethnographic literature of John Speth's, I love this quote because in talking about thinking of the meat that as it aged, made it, cut into it, and it was like the bursting of a boil, so full of great white maggots. He talks about his companion who eats them with evident relish. I being the ethnographer, so this Western ethnographer, criticized their taste, but they laughed at me and said, not illogically, you yourself like caribou meat. And what are these maggots but live caribou meat? They taste just the same as the meat and are refreshing to the mouth. I love this quote. And so in thinking about the importance of not just thinking about the postmortem interval application, but also thinking about, well, what about the consumption of these maggots in this putrid meat, kind of idea of these Paleolithic groups? Well, the maggots are kind of like the same idea as the muscle tissue. You have some animal, at some point it is going to attract maggots. Those maggots are going to have some hypothetical trophic level increase over the muscle tissue. I will tell you this does not work perfectly. In these entomological spaces, it's only about 1.5 per mil that I got in my experimental data, not the classic kind of 2 to 4 per mil that you would expect. But still, the idea is the same, is that here we have these dark dots are the same muscle tissue data, and these blowfly larvae, these are early decomp arrivals. So when the body is still quite fresh, quite wet. In this case, with this winter data, they arrived as early as February. So the bodies were placed at the end of January. These blowflies are arriving in February and are starting to be collected. But they're pretty stable. You don't see a lot of this increase in these maggots. These early decomp maggots, at least with this one pilot study that I'm looking at, don't seem to be contributing much to this increase in nitrogen value. I mean, they have a higher nitrogen value, but they themselves are not increasing over time. But the soldier flies. These guys are interesting. These soldier flies, they arrive as late decomp arrivals. So when we look at, again, based on these accumulated degree days, muscle tissue for the winter samples, they're placed in January. I was able to collect my last muscle tissue in April. These early decomp flies, their larvae are arriving in February, and they're kind of done by May. But in June, these guys arrive. And these are fascinating, in my mind, for forensic applications, as well as this idea of consumption of items that we don't think regularly on the paleo menu of these Paleolithic groups, which is the fact that these soldier fly larvae have such high values. If I go back really quick, these are these big nitrogens, so relative to that first muscle sample, these are in some cases in the mid-30s of their nitrogen value. That's huge. Like for a nitrogen person, that's obscene. Like even if you are like let's say, a Pacific Coast eating salmon, you're gonna get a signature somewhere in the neighborhood of, let's say, 20 per mil in nitrogen, high trophic level marine foods. The marine system has a longer food chain, so you get these really high nitrogen values. Even if you're in areas of Peru where there's evidence of like bat guano fertilizing the fields and you get these higher nitrogen values, still you're only in that same kind of like maybe 22, 23 per mil. That's above 35. Like, these raw values, I triple checked them because I had never seen values at 43 per mil. Those are the raw values, so not taking into account even the muscle tissue baseline that it's comparing to. These are huge values. This means that these summer versus winter maggots, they essentially are able to distinguish not on a regression line, an exact estimate of that postmortem interval, but say you have a skeleton that you come across in August, and you guys are in an area that snows, right? If someone dies in the winter, and then that decomp takes a really long time to progress, and they're skeletonized by August, well, was that skeleton that you found in August, is that someone who died and wasn't found for all those many months? Or is that someone who died in July and they skeletonized quickly and that decomp happened quickly? Well, these are drastically different values of soldier flies on the summer bodies versus the winter bodies. With a sectioning point of about 21 per mil, so right here, you get almost an 80% accuracy of distinguishing between a summer and a winter placement. I'm sorry, but that's pretty cool. Like, in terms of thinking about nitrogen, and this is just one application at one site for a
Are you
postmortem.
talking about nitrogen contents in the maggots?
Yes.
Or in the body?
The maggots themselves.
Okay.
So these are the nitrogen values. If you take that whole maggot, you collect them, I wash them, I freeze dry them, I grind them up so that they're all homogenized, the sample that I take is homogenized. And then these are the raw values that I get. So if you get that really, really high nitrogen value, you know, once you get into the 30s, you're like, there's no way that that's going to be a summer placement individual. So you can be really confident in kind of estimating what season of death you're looking at just based on these raw, so these are raw values. This doesn't even matter what the body, that baseline body value is. So it has a lot of potential from these two studies of looking at the muscle tissue and the maggots. There's a lot of potential for nitrogen to essentially be a conservative postmortem interval estimate. So we have this brand new method, and I can tell you from looking at 20 years of presentations at the American Academy of Forensic Science meeting, as well as the last 20 years of the American Entomological Society meeting, not a single person ever has used isotopes of fly larvae to do any kind of postmortem interval estimation. It is not something that anyone has looked at, and so is a wealth of potential to go into different regions, different time periods. I'm just showing you guys the first iteration of the first year of pilot study data. I have a whole second year that I placed individuals in earlier in the winter and later into the spring to look at these kind of shoulder seasons and how that changes as well.
Can I ask a quick question?
Yeah, absolutely.
So, but when you're getting just the dry bone, are you recovering the maggots in a forensic?
Yep. Yeah, so I've done one case now. There was one case that came to Tennessee that I was allowed, so case came in. I should actually follow up to see if that individual has been identified. And that individual was completely skeletonized and in a suitcase. And there were on her body 2 soldier flies within the suitcase. And so I did the values and I was able to estimate, say based on that maggot value that was likely a winter time of death for her. I should go back and see if they've actually, did they find out when she was missing, when she has died? I have not, this was, something I need to follow up on. But yeah, so you do. And in fact, that's the crazy thing about this study. You're a postdoc. You're trying to plan your schedule. You're like, I've planned this research study. I want to know when I can travel or when I can stop going and collecting every 4 days. Initially, I was collecting muscle tissue or insects. By later in the season, I was just collecting once a week the soldier flies. I was like, well, when can I stop? And they told me, they're like, oh, they're all soldier fly larvae, they're gone and dead by September-ish time period. I was collecting into December. As it turns out, these little suckers, they essentially hibernate. So they would be in the kind of decomp fluid-rich soil of the skeleton, either on the skeleton or within the soil that was just underneath the skeleton, in huge masses. So you're also thinking, like, if you're a carcass, you know, say somewhere in decompose, like these will collect and be in huge masses. Actually, soldier flies have been proposed as a great way for dairy farms to remove nitrogen, basically compost, because they are helping to consume and then remove waste. Like soldier flies are an incredible little entomological species. And so they, you know, so they would be in the ground. I'd be like, oh, they're dead. And I would collect one just to kind of make sure. And they'd be in my little vial in my hot little hand. And he'd come back to life. And he'd start to wiggle around. And I was like, that little bastard tricked me. They are alive. And so that was kind of the excitement and not knowing this. So yeah, I was able to collect these for a long time, which also, again, as I'm thinking about these Neanderthal Paleolithic individuals, you know, oh, you have a food source that's in masses, that's like a really high protein, high energy, you know, compact thing that you can just kind of scoop up, collect, and at least according to the ethnographies, are going to be refreshing to the mouth. So and there's a whole company that does maggot milk based on these soldier flies. You go down these, like, you learn fascinating things. Julie Lesnick, who's doing a lot of entomological work in human evolution and hominid evolution. She's fascinating and fun to chat with her about this kind of stuff. So we turn back to kind of this question of these Neanderthals, and is this a good study to approximate this? I kind of already indicated, you know, these are much larger species that they would be doing this potentially to. The donors that I had, you know, do not really maybe get on the low end of a reindeer. And so there definitely needs more work, but I would argue that the pilot information of muscle tissue decomp is there to suggest that this is a valuable hypothesis of John Speth's that he speculated, and that is likely a contribution that we're not considering. The other thing is to look at, well, you could say, well, Tennessee is not the environment that Neanderthals would have been living in. And this is a fair point as well, except that when you look at the actual temperature of the average high and the average low for Tennessee, based on this Smith et al. JHE paper that looks at a lot of these Neanderthal sites, that cold weather in Tennessee does actually fall within the range of some of the temperature data that you get at these Neanderthal sites. It is shockingly cold in Tennessee. I did not understand that as being a Californian and coming to Tennessee. It is shockingly cold. Not as cold as Indiana, not as cold as Wisconsin, but shockingly cold. And so what can we say about this consumption of putrid meat? Well, maybe. I would argue, based on this first iteration of data, that it is an interesting thing to think about, that it also could tell us about potentially the timing of when they were accessing this meat. Maybe they're only doing short periods of putrefaction, and you're not getting in that period where there's not much nitrogen change, and so it's not contributing to those high nitrogen values. Or maybe they're doing this much longer storage, and you are getting that much greater increase in the decomp meat, that putrid meat nitrogen values, and so that then is going to contribute to their higher nitrogen values. Probably no impact based on Boyer's work of the meat, but cooking in general, and especially if you have populations that are near the coast and they're cooking or boiling or roasting fish, there are a couple of per mil, and that's going to interpretively change your interpretation of those high nitrogen values. Oh, and then the maggots. I mean, how many maggots do you have to eat to raise your nitrogen value? Like, you know, we don't know, but the nitrogen values of maggots are so high that even if you're getting a little bit of that contribution, you know, say seasonally, at certain times of the year when those maggots have arrived and they're easy to collect, that potentially is going to have an impact to those nitrogen values. And so I think it's something to consider. And based on John Speth's proposal, this probably is something in the paleo menu that we're missing. And so for me, having this forensic anthropology project, but being able to turn from that data to kind of test this hypothesis of his is a very intriguing model that we don't often think about, kind of the dual aspects of the data that we can collect and the research projects we can construct to inform different areas of anthropology. With that, I always want to thank the donors first, definitely the Haslam Postdoctoral Fellowship, who funded all of this with their generosity of their postdoc funding, and then the various individuals who helped and volunteered collecting maggots and muscle tissues of graduate and undergraduate volunteers. So thank you, and I will take any questions.
So thank you very much. We have until 4:30 at the earliest for questions. So one, you have your own questions.
And I mean, we had some great questions during the talk, so I'm happy to entertain any further. Yeah?
Are there any extant human cultures that consume putrefied meat?
That consume putrefied meat? I mean, have you, I mean, sure. Have you ever gone and had like beef jerky essentially? I mean, like, at what level are you defining this putrefaction? Have you ever had a charcuterie board?
Yeah.
You know what I mean? Like,
Well, I'm thinking like rotten meat that consumes.
well, yeah, so
meat that we
Arctic
consider
groups
rotten.
still, Arctic potlatch and everything, so those same groups are still prepping that in this traditional way. I would argue that, you know, the minute it's not bloody and you're preparing it in some other way and you've processed it, you've dried it, like you are in this realm of processing in a food science sense of the term, that you're breaking down that meat in different ways, whether it's smoking, whether it's drying, whether it's this intentional whole carcass rotting of the meat. Yeah.
So do you think with Neanderthals, if we're going with this kind of traditional focus on big game hunting, then it would make sense to have it, because you're talking about having a surplus of meat.
Exactly.
And so ways of preserving it and storing it for longer periods of time that kind of go hand in hand, that we would see these other effects as well. Is that
Exactly.
a scenario?
Absolutely. And along with, you know, if anything, what the soldier fly shows me is this could go back even farther into thinking about scavenging of humans, of earlier hominins, when they're not hunting, but they're scavenging. And you're living in areas where this is another source of energy that they could be acquiring. And I can tell you, just from even modest sized human individuals, like, you would be able to sit and have a lovely little snack based on those insects,
Right.
that fly larva. Then of course you have the fact that you would have all other kinds of, you know, possible scavengers and birds and whatnot. But I mean, at Tennessee, there are birds there. They're not depleting this source to which it's hard to collect them. So yeah.
Cool.
Yeah. Oh yeah. This slide? Uh-huh.
So would number 5 be cultural processing of maggots?
Oh, would, like after that? Oh, absolutely. Yeah, I mean, cultural processing of food in general. I think that that's something that we're greatly missing in a lot of these paleo menu, is you have, you know, that cultural food processing. So I would define and draw that line at things like, how are you prepping the meat? Are you decomposing it as a whole carcass? Are you butchering it in some way and decomposing portions of it? Are you, you know, right there at the kill site? Are you, you know, setting up fires and smoking it and drying it in some way, doing some kind of preservation to that meat in some way? How is that then, you know, changing those values? You then go to that then actual, not the cultural, but the food prep. So you're cooking, you're roasting, you're boiling. So yeah, there's a graduate student who, well, she just defended her dissertation of Allison Brooks that further also looked at this. early iterations of her study, I would argue, had some different research design limitations. And so like she was looking at stakes and in the middle of Washington, D.C., putting stakes on the roof of the department building and exposing them to air and not seeing much change in the nitrogen. So, I think that as her stuff comes out, as I eventually publish, because I keep doing other things, and there was a pandemic, and so I'm late to publish the Neanderthal portion of this, but the maggot and the muscle and the maggot stuff are basically in the review process currently in a forensic journal. But as these things come out, there's going to be a lot more, like these are two studies, two different ways of looking at this, looking at whole body, looking at these cuts of meat. How are those changes happening? But regardless, I would argue that nobody's thinking about that before Royer did this study with cooking and thinking about how does that change. And honestly, the true tale in this and my true argument in this and what I presented on several times at SAAs and AABAs is that I don't think that we should be using mixing models to estimate paleo diet at all. I think that humans are too complicated, that all of these mixing models were built off of ecological species that had either 2 or 3 inputs of food, and they don't have that cultural overlay that humans have. And so I think I'm pretty negative about and pretty biased against these mixing models, because if you read some of these mixing model papers where they apply them to a population, they're like, for instance, they'll be like, the contribution of protein to the diet of, let's say, caribou meat was 25 to 75%. And you're like, cool, I could do a lot with that interpretation. So these mixing models have these really large ranges. And to me, I'm like, what really interpretively are we saying different about the diet than we already know that that's part of the diet from other zooarchaeological evidence? Like you can get at that from other ways. And I think that when you start with humans and you say you have so many dietary inputs, so many different in members already for these mixing models, and now you overlay cooking and food processing and invisible aspects to to that paleo menu, for me, it gets too complicated. For me, it just becomes, why are we using these mixing models? Are there any interpretive values that we're getting from them that we don't get from other lines of evidence? That's the thing that I'm working on and kind of get into very heated debates at conferences with people about, because some people love mixing models.
It'd be so exciting to see just how high you could get that 90 maggots by looking something else.
Exactly. But I mean, the hard thing with the maggot values. So if I went back to where my maggots are, okay. So if you're looking at like this, some of them are, and this is both the summer and the winter data together. So some of these values are quite high, and you get excited, but then I think, okay, well, in Central California, where you have salmon just rushing during spawning season, going up into their spawning area, there's salmon fish bone everywhere within these Central Valley sites, and so there's been an assumption in the Central Valley that they're eating and consuming a lot of salmon. Well, you get the isotope values, and you don't see the anadromous fish signature in the isotope value. So you start to have this, like, how much do you have to contribute to the diet to see that isotope value impacted? So I also wonder that with the maggots, we can see these super high values, but how often, like, is this snacking? Is this seasonal? If these late decomp guys are only arriving and they're not there for 6 months. So for 6 months, you got nothing. And for 6 months, you have the possibility of coming onto a carcass with a lot of these soldier fly maggots, how much contribution? So, I mean, like, that's where you start to get the, like, well, these are high values, but what are we really saying?
Any kind of handle on that?
No.
About,
absolutely not. Like, you'd have to do some super cool controlled feeding study that I can't even think of how one would.
But not even like animal models or anything?
I mean, they have these feeding studies, but nothing that I've seen that really would get to this level of contribution, because they're usually pretty simplistic with their animal chow mixes and whatnot. So if you were a fancier statistic person than I, maybe you could model it based off of the data that is out there, but I don't know. Yeah.
So I'd like to ask a simple-minded question.
No, none of those.
My understanding is, the original sort of findings, like Richard's, et cetera,
Right.
really compared Neanderthals to herbivores versus carnivores. And if you do that, it's pretty clear the nitrogen signal groups with the
carnivores.
right?
Yes, absolutely.
And so that, but, that's a very simple-minded idea of the Neanderthals as apex carnivores. I once saw Fred Smith give this talk, which changed my opinion. He talked about Neanderthal ethnology.
Right.
So once you understand that, then, and we have the SPY stuff, right? We have the dental calculus.
Right.
So then, okay, now we've moved from thinking of them in an animal model to thinking of them as a broad spectrum, modern hunter-gatherers, right? How broad we don't understand, but
Yes.
we have to change our understanding of that.
Absolutely.
So now I haven't read the most recent literature on the regional differences, but I understand that's starting to come out. So
Yeah, and not only that, but Clarvia, and again with Mike Richards, have done now single amino acid. So this is another way that you're not just looking at the bulk nitrogen, but you're looking at specific amino acids. And again, these types of methods, it goes back to looking at a lot of that meat, high trophic level carnivory that is definitely there. And I think that Mike, when he and I talk about this, he's not ever saying that there are really no plants, but just that is not, the focus. And so it was never about being opposed to, what Amanda Henry's work, has been doing. And when I was much more novice in stable isotope methods and coming from California, and then moving to my PhD, coming from the BioArc California data that I was working with, and moving into the PhD, reading a lot of this paleo work, reading a lot more of the Neanderthal work, I'm going like, well, what if you just looked at, these values and compared them? Now in that study, I did it like a paper for a class kind of thing. If you take all of these Neanderthal data that are published and you kind of average the baseline herbivore values to get kind of a like, okay, well, this is what the baseline herbivore value is. It actually basically overlaps with the California elk and deer population. So if you go like, all right, in California, where we know in prehistoric California, a lot more about the contribution of plant foods, a lot more about the processing of how they're processing foods. And then you look at the Neanderthal value, they're basically the same. They look like prehistoric Californians with essentially the same herbivore diet baseline. Now, in that study, I didn't take into context like different lab prep, different prep protocols, different labs running these values. But the great thing is, while that has a huge impact on oxygen values and why you can't easily compare oxygen values between different papers and different sites, carbon and nitrogen is a pretty stable workhorse, and a pretty stable workhorse no matter where you run. And so you do start to go like, well, we're calling these high, but they're only like 11, 12, like, are they really that high for humans?
But I would shift my focus from a single marker to thinking about regional variation, because now you're going to have expectations about changes in food preparation processes, right?
Yeah, and
so from northern populations to southern populations, you might be able to pick up some idea of how much post-processing there was in different Neanderthal groups.
Sure, and potentially yes, because here, we have, in France, you have Neanderthal is at 10 per mil. You have here, again, in Croatia, we have like around 11 per mil. You get a little bit, 11, 12. again, like 12, 13 per mil. So yeah, there are these differences in how high is high of the nitrogen value. I would hesitate, and because I always hesitate with over-interpreting what isotopes can tell you. So I think at least from the isotopes, we can talk about this to me in these broad kind of brush sweeps of saying, this is something we should be considering within the paleo menu. And this would be something that is making this a lot more complex. But to start to get at what you're talking about in these different areas of different cultural preparations, I think that goes way beyond what isotopes can tell you. And then you look to other types of evidence for that type of stuff, absolutely.
We have other types of evidence, right?
Yeah, no, absolutely. But, you know, we've also had isotopes for a long time until John Speth came along and kind of pulled this together in his retirement, you know. And the new iteration, he just presented at AABA's this year in a session that Herman Poncer pulled together. And he has a new monograph, but is looking at the tropics, same topic, but now the tropics. It's like 150 pages. I don't know where he's going to publish it, or if he's just going to publish it, you know, online and not worry about it, because he's like, I'm not cutting it down. I don't care. And that's one of the reasons it took so long for him to publish his 2017 paper and finally landed in Paleoanthropology, because they're the only ones who would publish it at the length that it was, and he refused to cut it. So you know, he was like, no, I'm including my 5 pages of ethnographic data of pull quotes that I've, you know, goes to support this. So, I mean, it's a beautiful article. It's a beautiful piece. So it has been an honor to be able to see his talk before it was published and then be able to be like, oh my gosh, I could do this in my postdoc. This would be awesome. And I mean, it was one of many projects that I did a lot of fingernails. I don't know how I ended up on fingernails and maggots. You know, I was always a bone and tooth person, and I was very happy in the bone and tooth world, but somehow I ended up in fingernail and maggot area for 2 years. And you sometimes stood there and you're like, where did my life take a turn? As you're like, you know, covered in PPE and you're collecting those samples. And I did find out, I'll tell you guys, I did find out there are lines of where I will not cross because I will not do it, of collecting maggots from certain places. Or I was just like, nope, not worth it for science. And one of my undergrads, I was at a conference and she was collecting for me and she's like, oh my God, I did this thing. And I was like, why? She's like, well, there was a maggot. And I was like, no, I would not have been able to do that. Like, I would never ask you to, I was like, no. So yeah, it was, there were some things where I was like, nope, that's my line. Like, I'll do this much for science and then I will not cross that. So yeah, it was a good time. Postdoc was a fun period for sure. Yeah.
I'm just a layman who lives in the neighborhood.
That's awesome.
I like to come to these things. If 2 individuals of the same species die at different times,
Okay.
as a long time passes, will their concentration of heavy nitrogen tend to converge?
That is a great question, and I'm going to go with no, because you said that they die at the same time.
No, die at different times.
They die
at different times.
Die at different
But
times.
then a long time passes. With the long passage of a long time, their heavy nitrogen isotope concentrations will tend to converge, don't you think?
That's an interesting thought experiment, because of course what I'm looking at is muscle tissue, and so I could only test the muscle tissue as long as there was muscle tissue to be tested. So there also seemed to be this kind of like it kind of peaked at like how much it would change. So in one sense, yes, there's only so much muscle on a body, kind of depending on what how much muscle you have. And that volatile compound process, that decomp is going to be pretty at a kind of steady rate. You know, so in some ways, yes, they will converge once they've lost their muscle tissue. If you're talking about like
fingernails and stuff like that?
So fingernails are pretty stable, as it turns out, and so kind of they do not seem to change over time, and they do not seem to change even for, let's say, up to 3 years of time is how much data I have from on the surface. Those fingernail nitrogen values don't seem to, there'll be a little bit of wiggle, but not an interpretive difference. And so nitrogen is pretty stable in certain body tissues, but in the muscle, in the flesh, it is going through this stage of decomposition. It is releasing this, ammonia and other nitrogen-based volatile compounds. So yeah, it's an interesting question. And then if you look at other tissues, say your bone, that's not going to change. That's also relatively stable. So that's not going to change based off of these. There are 2 different time periods, and they're converging because that's recording that baseline value for those individuals.
So what I'm driving at, my question is, if there is this convergence, then how do you really tell the time since death?
So, and that's a great question. So the muscle, this is where like having too many animations can drive you crazy. Okay, so the muscle tissue, and I'll do it on this one. So this is looking at these 10 individuals from the winter. And so when you're kind of like looking at that spread and that variation in that muscle tissue, you can see that when we're looking at this ADD, and this accumulated degree days is essentially how many degrees have impacted, okay, so we follow about that. So that's really common in forensic science to use that as a time marker to relate to. But you can see the regression isn't that tight. So there are other methods that use ADD, like it's something called total body score, and it has a much better regression than this. So you can get actual kind of estimations to like specific windows of accumulated degree days, but then you could relate back to the area where someone had died, and that's how they estimate a window of when they died. I don't think with isotope values that we have a tight enough regression to be able to do that specific of a time since death. And so what I think that it's better is that we can look at these broad general categories of, if you see this increase in nitrogen value, it probably is a winter placement. So there was a time of a long period of decomposition versus this short period of decomposition. And so I think that's where this interpretive value at this point of this method is.
In a jury trial, no judge would allow this evidence.
I don't know if you're correct, because it hasn't been tested. And I mean, it hasn't, so to date, isotopic data in general, I believe at this point, has been tested twice within a jury setting. So a lot of this type of data goes into broader aspects of evidence and are not often used as the sole thing of which people are testifying. And so though there have been cases that the person has been identified by their isotope data and their isotope profile, so for the biological profile, when that case goes to court, it's no longer a question of who the individual is, so there isn't someone getting up and testifying about that. So in that same vein, I mean, I agree with you, like we don't know how this would, currently this wouldn't pass because it hasn't been published, it's in the review process, right? But eventually, if it were ever to go to trial, one could turn to the peer-reviewed literature to meet standards of, rules of evidence and Daubert and all of this. So we don't know. But at this point, yes, it's preliminary. It's one site, it's Tennessee, it's one environment, but this is where I would argue the science starts, as well as for helping identify someone. And this is often best not in thinking of it identification, but methods to exclude people. So if you have an unknown human remains and you have a huge missing persons list, if you could take isotope data and say, well, that person definitely died in the winter, people that were still alive in the summer and had a different biological profile, you can exclude them. So isotopes are often used as exclusionary tools to narrow down, and then often don't make it into jury trials.
Okay.
That satisfies you though?
Yeah, okay.
All right, I win. It's a great question though, because you're right, isotopes have not been fully tested in the courts. And there's also, but where it has been tested is not in human, but in food science. So a lot of isotope forensics is actually based in food science. And so that's like things like people saying they've done it with like wine, with like cheese, with like honey. A lot of these products where people get really uppity and foodie-like about like things can only be labeled from certain places. And then you can look at, because all of this is, a lot of it's not nitrogen usually, it's carbon. So carbon signatures, oxygen signatures can give you what type of source, something is. A colleague, this is super funny, a colleague presented a poster on a couple of years ago, I guess, at the Super Bowl. There was like a beer commercial, and it was like, and I don't remember the specifics, it was like Bud Light said some disparaging thing about Coors and about where their beer came from. And there was an actual lawsuit that basically the one company brought against the other company of like, you have said like our beer is not what our beer is, but it actually is. And so she went and got the beers, tested the isotopes, and basically like showed this whole poster about this court case and how isotopes could refute or support the claims of the commercial from the Super Bowl that were being disputed in the court of law. So isotopes can be pretty fun in a lot of these forensic food areas. Yeah?
Yeah, this would be kind of culturally naive questions. It's like how live a culture life.
No naive questions. No, not at all.
But the data as regards Neanderthals, that the high nitrogen content is taken to mean that they're eating meat in advanced stages of decomposition. Is that the correct understanding?
Well, I don't think that we would ever be able to say that they're only eating meat in an advanced stage of decomposition. I think, again, that goes to kind of an overinterpretation of the values. But this is one more piece of the puzzle that when you're looking at bulk nitrogen values and amino acid specific, those compound specific amino acid or nitrogen values and amino acids, and you're looking at, the other final data and you're comparing all of these things, you could say like, well, they have really high values, but what's the contribution maybe of this food processing that they're doing that we haven't before considered? So it's more about is this possibly a contribution to this explanation of these high values? Is this cultural overlay of food processing that they're doing in some way? Are they cooking? Or, they have fire, are they cooking? Is that changing the values? Are they doing some kind of long-term storage like these modern Arctic populations? If they are, this could also contribute to these high nitrogen values.
As far as cultural practices surrounding the consumption of meat, ostensibly, are these the way that the meats are processed? Is the understanding there that these processing, the way that they process, are meant to offset the normal decomposition process?
Yes. So you're like, if you're say you're drying the meat or you're smoking the meat, you're doing that intentionally to not let it rot and putrefy in some other way.
Would that affect nitrogen contents as well?
My guess is yes. My guess is yes. So yeah, my guess is yes. I think that, where this all gets interesting is you have these different cultural ways in which people are preparing food. And like, Emily's talked about the fact that you could have these huge pills and you could have excess of meat. What are you going to do with that excess of meat? So then you're eating some things fresh, right? They're always going for the liver. You're eating some things fresh and not decomposing them, but other things could you be storing in these different ways?
Any parallel to current cultures? Are there any cultures that actually consume meat in advanced stages of decomposition?
Yeah, yes, you asked that previously. Yeah, these Arctic populations, people will still do those traditional food ways in very Arctic Inuit type of population.
And they don't try to offset the decomposition process by processing in a certain way, they just really just let it rot.
No, yeah, I mean, they have their cultural practice, and that's what happened in the 70s when Westerners were like, no, this is not health and safety. Health and safety standards are not here, let us give you some plastic. And so then there was this rise in botulism because they weren't doing it in their traditional foodway preparation. And so when that started to happen, they're like, no, thank you, we do not need your plastic containers that are supposedly healthier and safer, because they weren't, and went back to these traditional methods of food preparation.
Oh, and somehow these traditional methods of food preparation seems to offset any kind of the more pathological.
Right, they prevented things like botulism. They promoted that lactic acid bacteria, is promoting preservation of vitamin C, and you're getting, vitamin B12 that is accumulating essentially through this process of decomposition. So you're getting some extra vitamins, you're preserving some of those vitamins that are already in the meat. So yeah, they were doing it in these ways in which they could meet these nutritional needs that they had.
Are there any other questions? In which case, please join me in thanking our speakers.
Thank you so much for having me. I hope this inspires some of the students to think broadly about projects and, the multiple ways in which you can answer questions.